Hello, let's move forward with the second lecture of chemistry of carbohydrates. So the specific learning objectives for today's lecture are: first, you should be able to explain what are glycosides and its therapeutic importance, classification of oligosaccharides with their examples, describe disaccharides with their examples, and we will be looking specifically at lactose, maltose and sucrose, and lastly describe the biological importance of disaccharides with example. So let's move forward. First, what are glycosides? Okay, so glycosides basically are reactions between the hydroxy group of anomeric carbon, which is present in the hemiacetal structure of glucose, along with a phenolic or an alcoholic group or another monosaccharide. Okay, so to understand what are glycosides, first let's look at the structure of glucose. So this is the open structure of glucose which we have studied. Okay, so it's a 6 carbon having the aldehyde group, which is the functional group, which is present on the first carbon atom. Now this open structure basically physiologically in the human body, it is present as a ring structure, this format. Okay, so this is known as a Haworth formula. So how does this straight chain become a ring structure? It is because of sharing of the oxygen atom between the first carbon atom and the fifth carbon atom. Okay, so it forms a ring structure which is physiologically present. Now, depending upon the orientation of hydroxy group present along the functional carbon atom containing the functional group, that is the first carbon atom. Okay, so if the hydroxy group is present below the plane, it is called an alpha anomeric orientation. Okay, so the anomeric carbon atom which is present at the first carbon atom. Okay, if the OH group is below, it is called alpha. If the OH group is above, it is called beta structure. Okay, so this orientation of hydroxy group around the anomeric carbon atom is important. So when we talk about glycosides, we basically refer to this reaction of hydroxy group which is present in the anomeric carbon atom of the hemiacetal structure of glucose. So this hydroxy group can react either with another phenolic or alcoholic compound, or it may react with another monosaccharide. Okay, so this reaction is known as, this structure which is formed is known as glycosides. Okay, now if the let's say glucose reacts with another glucose, so another monosaccharide, so that's okay. If the attachment is, or the reaction is, let's say a non-sugar, okay, for example a phenolic or an alcoholic compound, so that non-sugar is called an aglycone. Okay, means not a carbohydrate. Okay, so the hydroxy group can react with another sugar, or it may react with a non-carbohydrate compound. Okay, so the bond which is formed is known as glycosidic bond. Now let's look at the glycoside which is of biomedical importance. The first one is digoxin. Okay, so digoxin is also known as a cardiac glycoside. Why is it called a cardiac glycoside? Because basically digoxin is a drug, okay, which improves the function of cardiac muscle. Okay, so if we look at digoxin, so this is basically the hydroxy group which is present in the hemiacetal structure of glucose. Okay, so this is the ring structure of glucose which we previously saw. Now this ring structure of this glucose is forming a glycosidic linkage with a steroid nucleus which also has a lactone moiety. Okay, so this whole component is basically reacting with our anomeric hydroxy group present on the anomeric carbon atom. Okay, so the hydroxy group which is present on anomeric carbon atom. So glycosides are basically a reaction of hydroxy group present on the anomeric carbon atom with, in this case it's a non-sugar. So this is the aglycone, okay, having the steroid nucleus and a lactose moiety. So this is the structure of a digoxin, which is a cardiac drug. Okay. Now what does this drug do? Okay, so this drug basically it inhibits the sodium potassium ATPase. Okay, so as a result of which this leads to increased contraction of cardiac muscle. Okay, so now sodium potassium ATPase basically sodium potassium ATPase channel, it maintains the concentration of potassium inside the cardiac muscles and sodium outside the cardiac muscles, basically the myocytes of the cardio of the cardiac cells. Okay, so normally we have is increased concentration of potassium inside and increased concentration of sodium outside. Okay, so the myocytes normally contain more potassium inside and more sodium outside. Now, if this is maintained by the sodium potassium ATPase pump. Okay, so basically sodium potassium ATPase pump is taking sodium from inside the cell and putting it outside, and taking potassium from outside the cell and putting it inside. So as a result, the concentration of potassium inside the cell is more and concentration of sodium outside the cell is more. Okay, this is normally the function of sodium potassium pump. Now this sodium potassium pump or sodium potassium ATPase is inhibited by this drug which is called digoxin. So as a result, the concentration of sodium inside the cell increases. Now this leads to secondary increase in concentration of calcium inside the heart muscle cells, okay, the myocytes, which eventually leads to increased contraction of cardiac muscle. Okay, this in turn is useful in case of heart failure. If the heart is not pumping adequately, is not functioning adequately, so in that case the contraction of the heart is increased. Okay, so this is a drug which increases the contraction of heart. So this drug is used in case of heart failure. Next is the next glycoside of interest is fluorescein. So fluorescein is basically a glycoside which contains sugar and a glycone which is called fluoretin. This fluoretin inhibits the sodium glucose linked transporters. Okay, so sodium glucose linked transporters are basically transporters in which sodium and glucose are together they are absorbed. Okay, so this SGLT transporters are basically located in the intestine, where sodium and glucose are together absorbed from the body after taking food, and they are also located in the kidneys. Okay, where again sodium and glucose are reabsorbed from the glomerulus. Okay, so when there is inhibition of sodium glucose linked transporters, then what happens is the amount of sugar which we are taking, the absorption of sugar into the system is inhibited, so the blood glucose levels decrease. Also in the kidney, what happens is normally this will cause increased reabsorption of sodium and glucose. Okay, so in case if we inhibit this, then the reabsorption of glucose also is inhibited. So as a result, there is loss of glucose in the urine. Okay, so this again leads to decrease in blood glucose. So this is an earlier prototype of SGLT inhibitors. Okay, now newer drugs have come. Okay, so this is this drug is useful for diabetics. Okay. The another glycoside of importance is an antibiotic called streptomycin. So streptomycin basically is aminoglycoside. Okay, so this is a glycoside having an amino group. So this inhibits basically protein synthesis in the bacteria. Okay, if the protein synthesis is inhibited, that leads to the lysis of bacteria. Okay, so it's an antibiotic, okay, which is used for bacterial infections. So these are the few glycosides which have biomedical importance. Let's look at the classification of oligosaccharides. So oligosaccharides are again of disaccharides. Examples are the lactose, maltose, isomaltose, sucrose. So lactose is made up of glucose and galactose. Okay, it is present in milk sugar and it is a reducing type of disaccharide. Next is maltose, which is made up of glucose and glucose. So maltose is a product of starch hydrolysis, and maltose forms osazone, okay, which is a star-shaped osazone, and maltose also is a reducing type of disaccharide. Isomaltose also is made up of glucose and glucose, okay, but the bond formation of isomaltose is 1,6, okay, between these two glucose molecules, and in case of maltose it is 1,4. So isomaltose also is a product of starch hydrolysis, and this is also a reducing disaccharide. The last one is sucrose, which is made up of glucose and fructose. Okay, so sucrose is present as in the form of table sugar, sugarcane juice, and sucrose is a non-reducing disaccharide. Okay, why is it non-reducing? Because the glucose and fructose the functional groups are in bond, so the functional groups are utilized. The functional groups are not free. So as a result, sucrose is non-reducing disaccharide. Let's first look at as lactose. Lactose is a milk sugar. Okay, so lactose is made up of glucose and galactose. So you have galactose over here and glucose. So you have one 4-glycosidic bond which connects galactose and glucose. So the functional group which is present in the first carbon atom, which is the aldehyde functional group, so that involves the bond with the hydroxy group of the fourth carbon atom. So if you look over here, the first carbon atom is here again, it is free. Okay, so that is the reason why it is reducing in nature, because the functional group present on the glucose carbon atom, okay, which is C1, the functional group is free at glucose. So this functional group acts as a reducing agent. Okay, so that is why lactose is a reducing disaccharide. Okay, the bond is 1,4. Okay, the first carbon atom along with the fourth carbon atom, the first carbon atom of galactose and the fourth carbon atom of glucose. Now lactose forms osazone, which is, it's a hedgehog or a powder puff shaped osazone. Okay. Now lactose is associated with lactose intolerance, okay, the symptoms of which is flatulence, bloating, diarrhea, and cramps, stomach pain. So lactose intolerance occurs because lactose, when ingested, is not metabolized by the body. Now why is lactose not metabolized? Because the enzyme which is required to metabolize lactose is not present, which is lactase. Okay, now if lactose is not metabolized, then lactose accumulates and lactose is converted to lactic acid. Now this lactic acid brings about all these symptoms: cramps, diarrhea, bloating, gas formation. So that is because of accumulation of lactic acid, okay, secondary to lactose intolerance. Okay. Now let's look at maltose. Maltose is made up of two sugar units, both are glucose. So here again the bond is 1 and 4. The first carbon atom of one glucose binds with the fourth carbon atom of the next glucose. So if you look at the second glucose here at the first carbon atom, here again the functional group is free. Hence maltose also is a reducing sugar. It's a product of starch hydrolysis, and on osazone, maltose forms a petal shaped or a star shaped osazone. Lastly, we have sucrose. Okay, sucrose is commonly present as table sugar or sugarcane juice. It is formed between glucose and fructose. The first carbon atom of glucose, which contains the aldehyde group, binds with the second carbon atom of fructose, which contains the keto group. So both functional groups which are present are occupied in bond formation. Hence sucrose is a non-reducing disaccharide, okay, because the aldehyde group which is present in the first carbon atom of glucose binds with the keto group which is present on the second carbon atom of fructose. So it is non-reducing, hence it does not form osazone crystals. Now we have a sucrose specific test. What is sucrose specific test? Sucrose specific test is a test which is specific for sucrose, as the name suggests. So here we take a sucrose solution and we perform Benedict's test, which is a test for reducing sugar. Okay, so the principle of Benedict's test is that reducing sugars, they form an intermediate called enediol, as we have seen in the previous lecture. So it forms an intermediate called enediol, which acts as a strong reducing agent. Okay, so this enediol is formed because of reducing sugar. Now since sucrose is a non-reducing sugar, so the intermediate enediol is not formed. Hence the Benedict's test is negative. However, if we hydrolyze the sucrose and we break it into individual glucose and fructose, then glucose also becomes a reducing sugar and fructose also becomes a reducing sugar. So then if you do a Benedict's test, you will get Benedict's positive. So an initial carbohydrate solution which is non-reducing, on hydrolysis it becomes reducing, is nothing else but sucrose. Hence it is called a sucrose specific test. Okay, so to summarize, we talked about glycosides. Because what are glycosides? Glycosides basically is a reaction of hydroxy group which is present on the anomeric carbon atom with another sugar or a phenolic or an alcoholic compound. The important disaccharides, the important glycosides which we discussed are digoxin, okay, which is a sodium potassium ATPase inhibitor, okay, which increases the force of contraction of heart, and it is used in heart failure. Okay, next was we talked about ferritin, okay, which is a sodium glucose link transporter inhibitor, okay, and it is useful in diabetics. And we talked about streptomycin, which is an antibiotic. Then we talked about classification of oligosaccharides as lactose, maltose, and sucrose. Okay, so lactose is basically made up of glucose and galactose. It is present in milk sugar. It is a reducing sugar, and it is associated with lactose intolerance in case if there is deficiency of enzyme lactase. So lactose is not metabolized, and lactose then accumulates and forms lactic acid, which brings about symptoms. We talked about maltose, which is made up of 2 glucose units, and it forms sucrose, which is star shaped. And lastly, we talked about sucrose, which is made up of glucose and fructose. The first carbon atom of glucose reacts with the second carbon atom of fructose, so both the functional groups are occupied in bond formation. Hence, sucrose is a non-reducing disaccharide. Okay, so thank you very much, and we will move forward with the third lecture of chemistry of carbohydrate. Okay, thank you.